A very warm greeting to all of you. Firstly, I would like to thank you, all of you, for joining us on the second day webinar of GlobalSeaweedSTAR, sharing best practices for seaweed aquaculture industry in Malaysia. Okay, before we start the scientific presentation, there are a few housekeeping announcements that I would like to remind all of us here. Okay, for all speakers, allow me to gently remind all speakers to mute your microphone unless you wish to speak. Okay, and to all participants, please use the Q&A box to type in your question and do indicate to whom your question is directed to. The question will be answered at the Q&A session after all presentations are being made. If you would like to receive a certificate of participant to this webinar, please sign up using the link in the chat box, or you can email to request through the SBP Malaysia email account. At the same time, please take note that this event is being recorded and will be shared publicly after the event on the GlobalSeaweedSTAR website, as well as social media account. If you do not wish for your personal information to be shared with other viewers during the event or appear in the recording, please join this event anonymously and do not provide any personal detail in the Q&A session. So without further ado, I think we will start our first talk today, okay, which will be presented by Dr. Pong Ziwan from University of Malaya on the title, Pests and Diseases in Malaysia, Eukematoid Farming. The floor is yours, Dr. Pong.
Hello everyone, my name is Ziwan Poon, and today I'll be sharing some of the key results from our work on the pests and diseases in Malaysia's eukematoid farming. For the benefit of those in the audience who are not familiar with the term eukematoid, it generally refers to the tropical red seaweeds, Carpophycus and Eucheuma, which are commercially cultivated for the extraction of carrageenan. So these seaweeds are also known as carragenophytes, and we can extract Kappa carrageenan from Carpophycus and Iota carrageenan from Eucheuma. So the difference is the gel produced will have different properties. Some also suggest that the term eukematoid should have a broader meaning to include other genera such as Betaphycus, which is cultivated in Hainan Island, and also Euchematopsis, which is largely cultivated in the Central America. In Malaysia, only Carpophycus and Eucheuma are being farmed. In terms of global production of eukematoid seaweeds, Malaysia currently ranks the third behind Indonesia and the Philippines. According to the statistics from the Department of Fisheries Malaysia, less than 100 kilometers square of sea area in Malaysia is being taken up for aquaculture purposes. But Malaysia's sea area actually comprises over 100,000 square kilometers. Until now, seaweed farming in Malaysia is largely concentrated in the east coast of Sabah. Thus, there is clearly a huge potential to expand seaweed farming beyond Sabah. This graph shows the production trend in Malaysia, and we can see that generally there is a decreasing trend in production since a peak in the year 2012. And this warrants immediate attention because seaweed farming provides a source of livelihood for over 1,000 farmers in Malaysia. The decline in production can be attributed to the same method of vegetative propagation being used for the past 50 years, whereby the plants are cut and reused as propagules or seedlings for replanting. And this resulted in clones and limited genetic diversity within the cultivars. This, along with changes in the environment and the climate, and also poor farm management, has led to increased incidence and severity of pests and diseases in the farm. In the next few slides, I will highlight some of the common pests and diseases in Malaysia's eucalyptus farming. The first one is ice syndrome, which is characterized by bleaching or loss of pigment on the seaweed, followed by degradation of the affected tissues. So ice has been reported to occur in all cultivated eucalyptus species, and seaweeds affected by ice suffer from loss of biomass from fragmentation. They also suffer from lower carrageenan yield and quality. And after an outbreak, there is also the issue of lack of new seedling or proper use for the next cycle of farming. So this potentially triggers a biosecurity crisis when farmers source for new seedlings indiscriminately and in the process brings in new pathogens into the surrounding water. So the ice syndrome is often associated with unfavorable changes in environmental conditions and infection by opportunistic pathogenic bacteria. However, until now, there is no single or combined causative agents have been definitely identified. For mitigation, farmers are advised to cut off the ice affected parts, which are then disposed of to land rather than in the water surrounding the farm. This is to avoid spreading of the disease to other parts of the seaweed or to neighboring seaweeds. From the small-scale sentinel farm study that our team conducted in 2019, Carboficus malaceus was found to be more susceptible to ice syndrome and also epiphyte infestation compared to Carboficus alvarezi. Next is the fouling algae, which are commonly found on the cultivation lines and also entangling the seaweeds. So these fouling algae compete with the eucalyptus seaweeds for light, nutrients, space, and even air circulation. Reports have also indicated that photosynthetic efficiency of the crops are affected. In general, less fouling is observed when the current is strong. Therefore, farmers should regularly remove these fouling algae from the lines and discard them on land instead of throwing them away in the sea. This will help to ensure better growth of the seaweeds, especially when there are no strong currents to wash them away. So in this study, we tease out the fouling algae from the cultivation lines for molecular identification, and the results indicate a wide diversity of species, including species of Hypnea, Laurencia, Acanthosiphonia, Acanthophora, and many others. So what I wanted to highlight is this Acanthosiphonia echinata, which was previously placed in the genus Polysiphonia and Neosiphonia. So this species was first reported in North America, and more recently it was believed to be introduced into Indonesia from the ballast water and hull fouling of ships from the Western Atlantic. So our results show that this species from the Western Atlantic is also introduced into Malaysia. Biofilms are also a common sight in eucalyptus farms. So this happens when silt and sediments from the seawater becomes deposited on the surface of the seaweeds, especially during periods of slow water current. These biofilms may decrease light absorption and harbor pathogens which induce ice syndrome and secondary microbial infection. Farmers are advised to manually shake the cultivation lines to get rid of these biofilms in order to prevent disease occurrence. Some of the common grazers reported in Sabah include turtles and rabbitfish. Grazing leads to open wounds on the surfaces of the seaweed and may increase susceptibility to ice and microbial infection. Turtles are particularly a concern for farmers as their flippers tend to damage cultivation lines and cause huge losses, as you can see in this photo. There has been suggestions or recommendations that turtles from conservation programs are to be released away from farming sites, and these calls for proper marine spatial planning. Innovative designs of cages and nets have been reported to be effective against grazers in seaweed farms in Indonesia. Studies have shown that epiphyte infestation decreases carrageenan yield and quality, leave black spots on the seaweeds when dry, and this resulted in lower selling price. The epiphytic filamentous braid algae penetrate into the surface of the seaweeds and they anchor into the host tissues using their rhizoids. This gives a hairy appearance. The point of entry of these epiphytes increases susceptibility to secondary microbial infection, and it also affects photosynthesis. Eucuma denticulatum has been reported to be more resistant to epiphyte infestation as it has more compact surface cells. Farmers should quarantine new propagules to check for epiphyte infection before growing out, and they should not use infected propagules, even if the infection is mild. This is to prevent the spread of infection. Prior to this study, EFA identification is solely based on microscopic examination. Through the use of DNA sequencing, we can confirm that the most common epiphytes in eupomatoid farms are species of Melanotemnus, followed by Ceramium, Chondria, and others such as Galiella and Lobelia junglemonoides. EFAs are also found on wild eupomatoids, although the diversity and density are less compared to their farm counterparts. We observe similar diversity of epiphytes between the farm and the wild eupomatoids, but one of the epiphyte was only found on wild host. The results of this study differ from those reported between 2003 and 2006, whereby the dominant EFA reported was Melanotemnus sabatieri and Melanotemnus apiculatus. Before I wrap up, I would just like to share this revolutionary idea of submersible cages with all of you, but I believe this would require large investments, especially from the big players of the industry. Some take-home messages include using healthy propagules, to always practice on-farm biosecurity measures, and to develop disease-resilient cultivars. So we will learn more about this from Dr. Tanji's talk later. And I would like to end my talk by thanking the colleagues involved in this project.
Okay, thank you, Dr. Pong, for such an inspiration talk and also share with us the latest finding resulting for the study of TWP1 on the pests and diseases in Malaysia eupomatoid species. And actually, there are many more new information that seen from what we have known before. Okay, thank you for that. So next, we would like to move on to second talk for today, which is by Dr. Tanji from University Tunku Abdul Rahman. He will share with us the topic on genetic diversity of Malaysian eupomatoid. The floor is yours, Dr. Tan.
Good morning and good afternoon, ladies and gentlemen. My name is Tanji from University Tunku Abdul Rahman, Perak, Malaysia. I'm an early career researcher under the GlobalSeaweedSTAR Work Package 3, which emphasizes on characterization, conservation, and exploitation of algal genetic resources towards crop improvement. Today, I'm very happy to share with you the findings of our project entitled Genetic Diversity of Malaysian Eupomatoids. The eupomatoids Carpophycus and Eucuma are important sources for the phycocolloid carrageenan. Owing to its gelling, thickening, and emulsifying properties, carrageenan is highly desirable by the food and cosmetic industries. Today, the farming of these red seaweeds is an important means of livelihood for coastal communities in Sabah, and this is being recognized by the Malaysian government. Malaysia is currently the third largest producer of carrageenan after Indonesia and the Philippines. Despite the commercial viability of eucalyptus farming, seaweed production in Malaysia has unfortunately recorded a general decline since 2012, and this decline may be attributed to various factors including climate change, lack of downstream marketing, poor farm management, pests and diseases, competitions, lack of coordination between stakeholders, and aging cultivars, which we will talk about today. The commercial eucalyptus cultivars of today are the same as those introduced since the 1970s. This is because they have merely been vegetative propagated over the years, which also means that the resulting propagules are all clones of the original. How is this a bad thing, you may ask? Since all these propagules are clones, if a cultivar is unable to adapt to environmental changes or new types of pests and diseases, there is always the risk that the clones of the same cultivar will all be wiped out. Also, Muller's ratchet states that in the absence of sexual recombination, which is the case for seaweed propagation, irreversible deleterious mutations may accumulate and lead to the decline of clones. So pretty much genetic decay. This is the reason why in mushroom farming, the mother culture is often preserved in glycerol at very low temperatures to prevent possible genetic decay. This is also the reason why agriculturists constantly perform selective breeding to improve plant cultivars, or set up seed banks for the preservation of germplasm. In the case of eucalyptoids, there's still a lot to do before good cultivar selections can be performed in Malaysia. One of these is to actually document the diversity of eucalyptoids that we actually have here in the country. Documenting the bio and genetic diversity of eucalyptoids in Malaysia will aid in conservation efforts and also identify valuable germplasm, the first step towards cultivar selection. The diversity of farm and wild eucalyptoids in Malaysia has been briefly studied by Tan et al., as well as Lim and co-workers 2014. The identification of seaweeds is usually performed using DNA markers, especially the mitochondrial encoded COX-1 and COX-2,3 spacer markers. The present study expands on this research by extending the area of sampling. The research methodology for this project is pretty straightforward. Cultivars were collected from seaweed farms from around Kuna, Sempona, and Tawau, whereas wild specimens, indicated by the red circles here, were collected from areas surrounding Pulau Bumbum, where they are known to be present. Specimens were preserved as vouchers. This is followed by DNA extraction and PCR amplification of the long COX-1 gene and COX-2,3 spacer. The success of PCR amplification was verified using geoelectrophoresis, and the PCR amplicons were then purified and sent for DNA sequencing. DNA contigs were assembled using ChromaSpro, and multiple sequence alignment were constructed and processed using bioedit. Molecular identification was performed using BLAST-N, as well as tree-based methods. Since we're interested in the genetic diversity of the eucalyptoids, haplotype networks were constructed using TCS. Moving on to the results and discussion. In terms of cultivars, a total of 37 Carpophicus elriezii, 30 Carpophicus striatus, and 7 Carpophicus malaysianus was collected. It was observed that Eucuma denticulatum was no longer being farmed in Malaysia. As for the wild specimens, a total of 43 Carpophicus striatus and 127 Carpophicus malaysianus were collected, out of which 23% of them are cystocarpic. On the other hand, 24 specimens of Eucuma denticulatum were collected as well. However, no specimens of Carpophicus elriezii were collected during this project. Now, before we look at the results obtained from this project, let us recap the valuable information generated by Lim et al. 2014, who genetically characterized the number of wild and farmed Carpophicus and Eucuma in Southeast Asia. According to the haplotype network shown here, which is based on the concatenated COX-2,3 spacer COX-1 dataset, 4 species of eucalyptoids, namely Carpophicus elriezii, highlighted blue, Carpophicus striatus, highlighted yellow, Carpophicus malaysianus, highlighted green, and Eucuma denticulatum, highlighted pink, were farmed in Malaysia back in 2014. These 4 species should also be present as wild populations in Malaysia, although the collection of Carpophicus elriezii was not successful, despite Carrageenan Island, Sabah, being the type locality of the species. The results from Lim et al. Also showed relatively large intraspecific genetic difference among haplotypes of Carpophycus striatus as well as Eucuma denticulatum. Next, DNA analysis of the farm specimens revealed that little has changed since 2014 in terms of the cultivars being used in Malaysia. Only one new haplotype, LBT10, was identified out of all the specimens. This could indicate the possible introduction of wild specimens as cultivars by farmers, or perhaps spontaneous mutation in a non-coding COX-2-3 spacer. The results has shown so far 2 haplotypes of Carpophycus alvarezii, 2 haplotypes of Carpophycus striatus, 1 haplotype of Carpophycus malaysianus, and 3 haplotypes of Eucuma denticulatum have been farmed in Malaysia. This also indicates the very limited genetic diversity within cultivars. The total number of global haplotypes based on the COX-2-3 spacer COX-1 dataset here is 7 for Carpophycus alvarezii, 8 for Carpophycus striatus, 4 for Carpophycus malaysianus, and 8 for Eucuma denticulatum. However, it should be noted that this number is an underestimation, as new and novel haplotypes have been recorded in the Philippines using other genetic markers in the past few years. On the other hand, the genetic characterization of wild specimens from Sabah, Malaysia, have revealed 6 new haplotypes for Carpophycus striatus, 15 new haplotypes for Carpophycus malaysianus, and 1 new haplotype for Eucuma denticulatum. Most of these haplotypes were revealed by the long COX-1 marker. The majority of the novel haplotypes of Carpophycus striatus appeared to be closely related to the KSB haplotype, which is shown here. And this haplotype is very different from the commercial Carpophycus striatus cultivar KSA. While the PL3 haplotype of Eucuma denticulatum has a closer affinity to the Endong variety of the Philippines or the Cacing variety that used to be farmed in Malaysia. The rich haplotype diversity of Carpophycus striatus, Carpophycus malaysianus, and Eucuma denticulatum around the seas of Semporna stresses the need to conserve the wild population of this species. Alright, to recap the highlights of this project, the eukymatoid cultivars in East Malaysia remain largely the same since 2014, and the haplotype codes designated for each species cultivars is shown in this table here. The limited genetic diversity of these commercial cultivars is not new nor surprising, as propagules have been commercially introduced for culture in various countries, but because of this, it is important that new ones are continuously developed to improve the resilience of the global eukymatoid industry. It is unfortunate that Eucuma denticulatum is no longer farmed in Malaysia, likely due to the unstable price and demand. However, results have shown that these species tend to be more tolerant of diseases and epiphytes and should be considered for reintroduction when the value chain is improved. The study on wild specimens has revealed a rich genetic diversity of wild Carpophycus striatus and Carpophycus malaysianus of Semporna, with more haplotypes likely to be discovered from less visited areas. These genotypes could serve as potential germplasm for cultivar selection and development further down the line. It is therefore important to expand the area of investigation and step up efforts in marine conservation, considering some of the wild populations were not within protected areas. Wild specimens of Carpophycus alvarezii continue to be elusive despite years of search. Although novel haplotypes which can be developed as cultivars have been discovered in the Philippines, extra consideration should be taken into account to avoid potential bioinvasion issues which may affect local populations of seaweeds should we decide to introduce these new cultivars. Also, the wild Eucuma denticulatum haplotypes collected in Malaysia differs from the commercial spinosome cultivar and could be investigated further. Although not elaborated due to time constraints, a few cultivar haplotypes were actually detected among wild populations in this project, which may indicate potential issues with escaped seaweeds from farms. Although these seaweeds were not cystocarpic, further studies should be done to ensure that this is truly the case. Lastly, future recommendations. Currently, Malaysia's seaweed industry could benefit most from an improvement in the seaweed value chain, but that is likely a topic for another time. In terms of genetic diversity of eukymatoids, the present study has offered a comprehensive documentation of farm and wild specimens, and in the latter case, for the very first time in Malaysia. However, there are likely more species or haplotypes to be discovered among the wild populations around Semporna, Sabah, which of course includes those of Carpophycus alvarezii. More importantly, The establishment of genetic documentation or even marker-assisted selection technology is only partial to the ability to culture and conserve germplasm long-term. In my humble opinion, it is important to be able to reliably culture and complete the life cycle of Kappa ficus or Eucuma, either in situ or ex situ, so that cultivar selection or artificial selection can be performed eventually in Malaysia. This was shown to be possible earlier this year by the Philippines, which is a very commendable achievement. The strive towards cultivar selection and improvement is an area that requires knowledge, talent, commitment, collaboration, as well as funding. And despite the many challenges, this is an area of great importance if we are to propel the global seaweed industry to greater heights. With that, I would like to end this presentation for the project supported by GlobalSeaweedSTAR. Thank you very much for your time and attention.
Thank you, Dr. Tanji, for sharing the latest update as regards about the genetic diversity of eucheumatoid, both farm as well as wild eucheumatoid. So as we know that with the funding that given by GlobalSeaweedSTAR, this has allowed us to have the very first kind of such an intensive searching for the wild population. And through the study that we have found that Malaysia coastal water, especially in the state of Sabah, that we have the wild cultivar that can potentially be developed to be a new cultivar. Okay, with that now, I would like to move on to today, the third talk, which is by Dr. Cecilia Cambe from University of Malaya. She will share with us the topic on biosecurity application in improving the productivity of eucheumatoid farming. So, Dr. Cecilia, the floor is yours.
Hi everyone, I'm Cecilia Cambe. In this sharing best practice webinar, I'm going to present biosecurity application in improving productivity of eucheumatoid farming as 3 years research on biosecurity in seaweed aquaculture. About concept and its application, why biosecurity is important to be used in seaweed aquaculture industry. In this study, concept of biosecurity based on definition given by Food Agriculture Organization cover to food safety, introduction of animal, plant, disease and pests, introduction of living modified organism, and introduction and managing the invasive alien species. So based on the concept, biosecurity application adopted in seaweed aquaculture is suitable to manage disease outbreak, increase product quality, improve environment support system, which all for sustainability of the industry. Why biosecurity application is needed to adopt in seaweed aquaculture industry in Malaysia? The reason is there has reduction in seaweed aquaculture production from 2012 to 2018. The ice syndrome was experienced by 50% of farmers in 3 most productive regions. Uncontrol of infestation of epiendophyte filamentous red algae through imported and translocated seedling, which can cause disease and low quality of the seaweed. However, strong policy and regulation have framed the seaweed aquaculture industry in Malaysia. So based on the challenges, we want to explore and study the condition of seaweed aquaculture in Malaysia using biosecurity approach. So the purpose of GlobalSeaweedSTAR biosecurity research were to identify the biosecurity concept adopted in Malaysia national policy and regulation, to evaluate implementation of the adopted biosecurity in policy regulation to the farmer as the main stakeholder of the industry, to assess farmers' knowledge, attitude, and practice toward biosecurity in managing disease and pests in their farm and other farm risks, to assess effectiveness of biosecurity application on farm level as the core production system of the seaweed aquaculture. First study aims to identify the biosecurity concept that apply in the national policy and regulation. There are 4 policy and regulation that often used in managing in seaweed aquaculture in Malaysia, which are Malaysian Standard Number 2467 produced in 2012, Fisheries Act 1985, Malaysia Good Agriculture Practice or MyGAP, and the Seaweed Farming Manual. We analyze this policy and regulation using the thematic analysis. Results show most of policy and regulation are non-legally binding, lack adopt precautionary principle, and limited use of biosecurity approach. But the good things of this regulation, they are using seaweed explicitly and information use based on scientific knowledge. Based on the result, we assume that the non-legally binding of most regulation, consequently, the regulation will be less uptake by farmer. Therefore, regular monitoring and evaluation by competent authority to the aquaculture system is needed to control the practice and risk level in whole farm system. Limited biosecurity approach and unuse of precautionary principle make the seaweed aquaculture system remain less resilient to the change. In the second study, we evaluate implementation of biosecurity adopted in the seaweed framework by assessing farmers and government officer knowledge, attitude, practice toward biosecurity. We surveyed 67 farmers and 10 government officers in 3 most productive regions during June to August and November 2019. In the questionnaire, we include the question about understanding of disease pests and its pathway, disease management, and general control on farm system. The KP results were cross correlation with the written context of the national policy and regulation that we observe. The result of KP study show that farmers have a fair index on knowledge toward biosecurity. The lack knowledge caused by lack understanding of parasite and invasive species among farmers, insufficient understanding on disease pathway through equipment, grazer, and epiphyte, and also uncontrolled in nursery area. The attitude of farmer also indicate fair, but on-farm biosecurity practice of farmer were found poor, and farmer in Sampurna and Kunak were had lower practice score. The insufficient practice found the no disinfection practice of farm equipment by most of farmer in Sampurna and Kunak, reuse of infected crop on farm, lack of crop quarantine, lack in crop management, and also lack in reporting system and farm management. The knowledge and attitude toward biosecurity of the government extension officer was also indicate fair, but they have good in biosecurity attitude. The result of KP cross-correlation with the national policy and regulation found lack implementation of biosecurity measure in practice due to no procedure for disinfection process, no procedure for crop quarantine or crop movement for disease prevention strategy, no practical guidance to handle infected crop, and no regulation concerning reportable of the farm system. Government officers also need to improve in biosecurity knowledge on disease and pest management to fully assist farmers in their farm. This result may confirm that majority of seaweed farmers came from small-scale individual farmers that have low awareness on managing their farm due to low education and as temporary job. The result of the KP study have published in Marine Policy Journal in 2021. The third study aims to assess effectiveness of biosecurity application on farm level. Observation was conducted in a small-scale farm for 40 days in Bumbum Island, Sampurna, Sabah during the period of peak disease season, which is June to August 2019. Evaluation of biosecurity measure on farm conducted by comparing the incidence of ice in Capapicus malassianus and Capapicus alvarezi at the farm with biosecurity measure and the farm without biosecurity measure. Treatment between farms were focused on detection on the health condition of propagule for whole period at the farm with biosecurity treatment only, and routine removing the pests every 2 days by cleaning all pest coverage on crop and rope at farm with biosecurity, while other farm was follow normal practice by farmer. Result of the biosecurity application on farm are shown by two graphs for Capapicus malaceatus. In graph A, the seaweed grow in non-biosecurity treatment farm. The coverage of pests were increased with increased incidence of ice reached to about 30%. In graph B, the seaweed grow in biosecurity treatment farm were had low coverage of pests with low incidence of ice. Removing the macroalgae epiphyte and biofilm waste can minimize the ice outbreak up to 75%, but the epiendophyte coverage cannot be removed easily unless proper detection on farm preparation or unuse of the infected crop can decrease the outbreak on farm. For Capapicus alvarezi in graph A, the coverage of pests increased with increase of ice incident, while graph B show decreasing the ice incident with low coverage of the pest. Removing the macroalgae epiphyte and biofilm waste caused reduction of ice up to 71%, and the Capapicus alvarezi was indicated as resistant to ice and the epiendophyte. We also measure the water temperature, salinity, inorganic nutrient, ammonium, and water current between farm. The results show that no significant difference of both farm, but the ice syndrome was occurred differently. It means cleaning treatment in biosecurity farm was given a significant effect in minimizing the ice syndrome. The result of this study also published in Journal of Applied Mycology 2021. As conclusion, biosecurity measure is important for a eukematoid farm, but challenging to be implemented to farm with low awareness to their farm and lack biosecurity knowledge. Supporting from the competent authority is needed to keep farmers to do proper biosecurity measure in practice and to encourage their knowledge in disease management. Need to have an adequate standard in aquaculture system, include the detailed procedure with fully concern to biosecurity concept on farm and at national level. This research was part of the GlobalSeaweedSTAR project funded by UK Research and Innovation and Global Challenge Research Fund. So thank you for your attention.
Thank you, Dr. Cecilia, for sharing your research on the biosecurity. And based on the result of your study, it shows that biosecurity does promise a very good, high potential in increasing and boosting the production of seaweed aquaculture. Okay, now we would like to move on to the fourth talk for today, which will be presented by Mr. Muhammad Azam Asri from University of Malaya. He will share with you the role of migrant in seaweed aquaculture in Malaysia. Mr. Azam, please. Thank you.
Assalamualaikum and hi everyone. I am Azam Asri from Malaysia. I am the early career researcher from work package 4 of GlobalSeaweedSTAR, which focus on the socio-economic of seaweed industry in Malaysia. I will present the impact of migrants on Malaysian seaweed production, which this is one of our findings from a study that we conducted over the past 3 years. This webinar that has been running for 2 days may help us to familiar with the type of seaweed that we discuss this time. My presentation focus on 2 types of seaweed that are cultivated for commercial purpose in Malaysia, namely Capapicus and Euchema. Both types of seaweed are cultivated commercially for the carrageenan production. In 2012, Malaysia produced around 300,000 tonnes of wet seaweed, and the value is around RM 200 million. But the production has been reduced to 188,000 tonnes of wet seaweed, and its value is around RM 65 million. Annual seaweed production has decreased by 50% when we compare from 2012 to 2019. Different factors can explain why seaweed production is decreasing, such as impact of disease on seaweed crops, traditional farming technique, price fluctuation, predation by herbivores such as fish and turtles, infrastructure damage following storms, or the unsuitable scale of operation. Several studies in Malaysia have addressed biological and macroeconomic aspects of seaweed cultivation, but fewer have focused on socio-economic issue. So this paper attempts to analyze the impact of migrant participants in seaweed production in Malaysia using a value chain analysis as the conceptual framework. In order to analyze the roles of migrant farmers play in the seaweed value chain, it is necessary to understand the complex nature of their citizenship. Migrants can be divided into legal and illegal migrants, and consisting of various forms of identity such as IAM 13 letter of birth by Sabah Chief Minister Department, census certificate by National Security, Sea Gypsy, or even the stateless. So this presentation will not discuss the complexity of the issue of citizenship, but provide an overview of the situation faced by the seaweed farmers who are not citizens. So the term migrant is used as an umbrella term to define a non-professional group of people that have in common, which is their lack of Malaysian citizenship and restricted access to any formal system like education, employment, or healthcare. In this study, I use value chain analysis as a research framework to answer the research question. And for the methodology, we are using mixed method, which is rely on key informant interviews, household survey, and observation. Fieldwork is conducted in 3 locations in Sabah, which is in Semporna, Tawau, and Kunak. As a result, the data collection that we have is we managed to interview 77 farmers, which is household survey, and this consists of 44 citizens and 33 migrants. And also we managed to interview about 24 key informant interviews, which consists of 2 collectors, 2 processors, 6 cooperative, 2 exporter, and also 12 government officer in Sabah. This diagram is the value chain that were developed through this study, which consists of value chain influencer, supporters, and players. In this presentation, attention should be given to the value chain player, which involve migrants in these activities. A value chain player consists upstream marketing and also downstream activities. Migrants are not officially own a farm or to participate in any government cooperative. They are therefore either independent, non-registered farmers or hired by citizen cultivators to farm as permanent or contract workers. Migrants are also involved as a collector or intermediaries. In general, many migrants are involved as independent farmers and also intermediaries. Migrants who participate as independent farmers and intermediaries can carry out their activities informally and do not receive aid directly. Next is the key findings of this study. The first one is about the rule and regulation on permission to farm. In the table that show in the slide is showing the cultivators and also the workers. Cultivators are those who cultivate seaweed and own a cultivation area. Cultivators are therefore citizens who are registered as cultivators with the government, where workers are the ones hired by the cultivators to cultivate the seaweed. Only migrants who have passport from their country of origin can have a special pass or temporary work pass and be hired legally by citizen. The rest of the migrant will be hired unofficially. This means that the official number of seaweed cultivators is likely to underestimate the actual number of seaweed farmers operating in Sabah. The second findings is about the rules and regulation on sea usage. Given that migrant farmers can cultivate unofficially and the migrant farmers area are not officially recorded due to their status, this affects the lack of enforcement in the formal rules and regulation that governing where seaweed can be produced. This has caused conflict between migrant seaweed farmers and other sea users cannot be reduced, such as seaweed trawling, fishing, or boat pathway. The last findings that will be discussed in this presentation are about the migrant contribution to the national production. It is not possible to present accurate data that differentiate between the citizen and also the migrant production. The figures that present in this presentation should not be taken as a migrant farmer's true contribution to the Malaysian seaweed production. However, this is the first time in any attempt has been made to recognize their overall contribution. To provide an initial assessment of the contribution made by the migrants, we provide an estimate of production volume from both citizen and also migrant farmers. Since the number of citizen farmers is known, their total production can be calculated and compared to the official statistic. The difference between the two figures can be assumed to have been produced by the migrant farmers. The calculation made in this study by assuming an average productivity of 1.06 ton per cycle, this is the highest productivity recorded by a seaweed cluster project participant. And the second one, we assume that 6 production cycle per year. So in the graph, the orange line is the production made by the citizen and the blue line is the official national production. The green area is the estimation of the seaweed production by the migrant or unregistered farmers. Migrant farmers could currently be producing over half of all the seaweed production in Malaysia. Next is the impact of migrants on Malaysian seaweed production. In this presentation, I will present two impacts. The first one is about the quality and yields of the seaweed, and the second one is the ill-informed policies. A migrant can legally access to working permit, farming aid, healthy seeds or training. And as in Malaysia, we have a Malaysian Good Agriculture Practice or MyGAP, which is the MyGAP has a limited reach as migrant farmers are often unaware of it. So this will cause a migrant to have less knowledge regarding biosecurity, and their production is vulnerable to disease and predators. As a migrant and unregistered farmers represent almost 50% of the national production, their lack of information on biosecurity can reduce the quality and yields of Malaysian seaweed as a whole. Ultimately, this leads to migrant farmers leaving the seaweed industry and will affect the volume of a national production. And the next impact is the ill-informed policy, which is the migrant actively participate in the seaweed cultivation activities informally. Informal activity such as the number of cultivators from them or the number of their seaweed farms cannot be officially recorded. So the absence of the accurate and precise data concerning seaweed cultivator may contribute to ill-informed policy and initiative concerning the welfare and the development of coastal community and also the seaweed sector as a whole. For the conclusion and recommendation, this study has highlighted the central role of migrant farmers that they play in the seaweed cultivation in Malaysia. Both citizen and migrant farmers acknowledge that the formal and informal arrangement between them are actually mutually beneficial and contribute to maintaining the seaweed production. However, migrants face an institutional stumbling block that restrict their ability to participate fully in the seaweed industry. This affects their ability to secure their primary livelihood and improve their productivity. Recognizing the migrant as official farmers with a significant contribution to the seaweed production will help Malaysia to improve this industry. Integrating migrant production in monitoring information will not only improve the accuracy of official statistics, it will also build a more accurate picture of the full impact of a seaweed production on the local economy, particularly is in providing a source of income to the most marginalized member of coastal community. So further research could quantify more accurately the migrant contributions to seaweed production in Malaysia, which is leading to a better targeted policy that will help the government of Malaysia achieve its goal of a more sustainable, resilient and productive seaweed industry. So that's all from me about the migrants farmers in the seaweed cultivation activities. Thank you.
Okay, thank you, Mr. Azam, for sharing your finding based on mixed method approach that highlighting the importance and the contribution of migrant to seaweed aquaculture industry in Malaysia. Okay, so the 4 previous talk is actually given by all the early career researchers from Malaysia that share with you resulting from our 3 years involvement in carrying out research. Now we would like to move on to the second part of today's presentation, which will be brought by all the postdoctoral researchers from the UK institution, which their research finding or their talk will be mainly based on global perspective. Okay, for the 5th talk today, I would like to invite Dr. Georgia Watt from Natural History Museum of UK to share with us on the influence of ice disease on microbial community associated with cultivated eukymiotoid. Okay, Dr. Watt, please. Thank you.
I'm going to be talking today about ice disease in eukymiotoids, starting by focusing on some of the work that we've published as part of the GeoSTAR consortium more widely, and then specifically on the experimental work looking at the associated microbial communities with the Malaysian team. So we know the commercial cultivation of Capophycus and Eucheuma has increased rapidly since the industry began in the 1960s, and the greatest constraint to its further expansion is the occurrence of yield-limiting diseases. So these are epiphytic filamentous algae, like Melanothamnus, which is being covered separately by C1 and is shown at the top here, and also ice disease, which is this bleaching condition shown here, which was first seen in the Philippines in the 1970s, but has since become both prevalent and widespread. And it should also be noted that these conditions are known to co-occur within the same tissue as well. So far, there's no definitive cause of IID, though we know that it appears to affect all cultivated eukymiotoids, and it's been recorded in all regions where cultivation is either established or has been trialled. In order to gain a greater understanding of the research to date concerning IID and its associated causes, we worked on a review paper attempting to compile as much information as possible relating to the condition and placing it in the wider context of what's known about similar bleaching syndromes in other sessile marine holobionts, including red algae and corals. So we included as broad a range of literature in this as possible, both English and regional language journals and reports, and we also included as wide a range of sources as we could as well. So we included peer-reviewed journal articles, government and international reports, for example those from the FAO, and grey literature including academic theses and research institution reports. In particular, we were looking at the terms used to describe IID signs, so what are the commonalities and differences between reports. We also looked at what the attributed causes were and the methodologies that were employed to determine this. So ultimately, we were able to look critically at the state of research into IID. We identified a number of shortcomings, and we also identified a number of areas where research effort could and should be applied to elucidate the causes and mechanisms of disease. In the table that we compiled for the paper, we had a total of 57 reports, and unfortunately 23 of these gave no description whatsoever of the observed disease signs while discussing IID. And this is a problem because it limits how useful these studies can be for other researchers, because we can't state for certain whether the disease signs observed were rightfully attributed to IID or whether this was the occurrence of another macroscopically very similar condition. So almost all reports describing the initial presentation of IID included the terms bleaching, whitening, depigmentation or loss of pigmentation, in some cases stating that this occurred in white or pale spots. A very small number stated that this was preceded by the presence of a white powdery growth on the surface of the thalli. And not all reports included a description of the progression of the disease, but where they did, this largely reported the disintegration, degeneration or softening of the thallus, with others noting production of organic substances by affected tissue. And in a couple of reports, rather than a softening of tissue, there was this progressive hardening, which was noted, that was followed by degeneration and fracturing of the thallus. A range of causes of IID were given, which can be broadly categorised as abiotic, shown in green, biotic, shown in blue, or a combination of biotic and abiotic factors, shown in teal in the centre here. The most common single cause is the opportunistic infection or proliferation of bacteria. Where a study attempted to characterise these, often a putative Vibrio, Pseudomonas, or Cytophaga was identified. Bacterial infection was sometimes coupled with unfavourable conditions as a potential cause of IID, and a very small number of reports also implicated fungi as the cause of IID. As mentioned earlier, EFA were also linked to IID as a potential cause, either as a single factor on their own, as a precursor to bacterial infection, or in combination with environmental factors. In terms of abiotic causes, unfortunately, many reports are very vague and link IID only to environmental stress or unfavourable environmental conditions. Where factors were identified, this was most often a decrease in salinity paired with an increase in water temperature, and both of these were also reported as causative factors on their own too. So what did we learn from this? Well, the majority of studies which attempted to characterise the bacteria associated with IID use culture-based methods, and unfortunately we know that these can have some major biases and limitations. Firstly, many different growth media were used across these studies, making them impossible to compare in any meaningful way, though it should be noticed that most commonly the media used was selective for taxa like Vibrio, which may explain their apparent association with IID. Secondly, we know that the majority of marine bacteria can only be cultured under specialised conditions, with many others not able to be cultured at all. However, the biggest limitation of these studies is that they very rarely included any isolates from healthy eukymatoid tissue for comparison. And this is important because without them, you're unable to say whether the bacterial taxa you're finding in your diseased tissue are part of the normal microbiota associated with eukymatoids, and therefore you can't definitively associate them with the disease signs that you're seeing. Very recently, there's been a number of attempts of microbial sequencing, either using Sanger, which often relies on cultivation and therefore carries forward the same limitations, or Illumina high throughput technology, which I'll come back to later. But unfortunately, these studies fell foul of the same problem as mentioned before, and in most cases, they didn't include any healthy control material. So our sampling was completed 2 years ago in July 2019 in Malaysia, where we used a site in Sabah, which we refer to as our sentinel farm. So here we looked at Capophycus malaysianus and Capophycus alvarezi over a 45-day cultivation cycle on the same site, where we sampled at least every 9 days. The farm itself was monitored daily, and when disease signs were spotted, an extra sampling event took place. At every sampling event, a number of environmental parameters, including the water temperature and salinity, were recorded, and the team collected a total of 6 healthy plants, or up to 6 plants with IID at each point. These were very carefully photographed to record the gross pathology, and the tissues were collected from the apex, the secondary branch, and the primary branch. Where plants with IID were collected, healthy control tissues were also collected from the same individual. So these tissues were preserved in RNA later for DNA extraction and high-throughput sequencing, and also in Davidson solution and glutaraldehyde, so they were available for microscopy if we required that. Rather than bacterial cultivation, we choose to use high-throughput sequencing methodologies for our sentinel farm experiment because this offers a number of very clear advantages. So the first of these is that there isn't this bias towards cultivar microbes. The DNA extraction method that we used was specifically designed to completely homogenize and lyse all cells in the sample, so the total diversity would be sequenced. This includes taxa that are rarer and less abundant, and it also enables us to look at both prokaryote and eukaryote taxa in the samples. We used amplicon-based methods, which involve amplifying a short barcode read-in. This was a short section of the 16S ribosomal RNA gene for prokaryotes and the 18S gene for eukaryotes. And these are then classified against databases to identify the taxa present in the samples. There are a couple of drawbacks to amplicon-based methods, and that's that there's a small possibility that genetically divergent taxa present in the sample will not be amplified using these methods. And identification of taxa also relies on very well curated databases to accurately assign taxonomy. But what we're aiming for really is to enable the comparison of microbial communities associated with healthy and IID afflicted tissues. Going back to our sentinel farm experiment, we found that IID was observed at all time points after T0, so the day that the plants went out on the ropes, but was particularly prevalent at the T9 and the T18 time points, which I'll be focusing on in the preliminary results today. Generally, fewer IID affected tissues were available to be collected from the later time points, but this is not because the disease was less prevalent over time. Rather, as disease progressed, affected plants detached and were lost to the tide. And also we had a lot of problems with turtles grazing on our cultivation lines during the experiments. So looking at the measurements taken of the environmental parameters across the experiment, we can see that there was some fluctuation, particularly between the first day of the experiment and the first sample collection on T7, though it should be noted that, especially for the nutrient and pH measurements, these are actually very, very small and certainly well within normal ranges for coastal seawater. Here we have the bacterial communities from samples collected on day 9 on the left and day 18 on the right. So on the top row in each case are the IID samples, and on the bottom the equivalent healthy tissue. For day 9, we have IID tissues from apical and secondary branches and not the older primary branches, which we do have for the T18 time point. So there are differences between the healthy and diseased tissues at each time point, and there are also differences between the diseased tissues from the 2 time points. On day 9, the healthy tissues show a high relative abundance of Flavobacteriales in the green, Alteromonidales in this salmony orange colour, Rhizobiales in lilac here, seen in the secondary branches in particular, and Cyanococcales in bright pink. In comparison, the disease samples show a much higher relative abundance of Bacillales in this lighter orange and Celvibrionales in the green here. The healthy tissues from day 18 are similar to those from day 9, with the exception of Flavobacteriales, which is only abundant really in this single sample here. The relative abundance of Cyanococcales and Alteromodales is also increased in these samples in comparison to the earlier time point. So you can see the diseased tissues from day 18 have a higher relative abundance of Flavobacteriales, though this is not the same sequence variance as seen in the healthy tissue from day 9, and also a higher abundance of cell vibrio here, particularly in the secondary and primary branches. The relative abundance of bacillales in disease samples from this later time point is much lower as well than it was in the T9 samples. So if we think specifically about the taxa which have been previously associated with IID, we can see from the legend at the bottom that there are Vibrio and Cytophagales reeds in some of our samples. However, Vibrio only accounts for more than 6% of reeds in a single sample, either healthy or diseased, and that's this one here, this diseased sample from T9, which has 25% of reeds assigned to Vibrio. For Cytophagales, this is only present in a handful of the T9 IID samples and a handful of the T18 IID samples. It doesn't occur in any healthy tissues, and at neither time point does it occur for more than 5.8% of reeds. Looking at the eukaryote diversity, we can see again that there are clear differences between the healthy and diseased tissues. So the healthy tissues appear to be dominated by the ciliates Ephilotidae in the day 9 time point, and to a lesser extent the day 18 samples as well. We can see other sectorian ciliates such as Acinetidae in day 18, and what's actually interesting to note is the low but consistent relative abundance of Rhodomalacia and Alveoles in both the healthy and diseased tissues from both time points. So 18S is a fairly poor molecular marker for both of these groups, but the closest relatives to the sequence recovered are Neosiphonia and Strepocladia for Rhodomalaceae and Ulva for Alveoles, and so these are being sequenced separately using more informative markers, so that's RBCL and TAFE respectively. With the exception of Ephilotidae, the same eukaryote taxa are present in the diseased tissue, though at a much lower relative abundance. For the day 9 samples, the diseased tissues are dominated by rapid pinnate diatoms, so relatives of Nietzsche and Navicula, and Neoparamoeba and its endosymbiont Perkinsiella in the apical sample. For the day 18 samples, most of the diseased tissues are dominated by reeds assigned to Labyrinthulaceae and Masseteridae. So some labyrinthids are known to be parasitic, but we were unable to see any evidence of this in our microscopy. But we also know that other labyrinthids are saprophytic, and described Masseteria species are often associated with aquatic detritus. So it may well be that these taxa are present as opportunistic saprophytes, feeding on damaged eukymatoid tissue rather than being parasitic. We can see from the data that there are differences in prokaryote and eukaryote microbial communities, not only between healthy and eye-sized diseased tissues, but also between time points during our Sentinel farm sampling. Now, this is really interesting, and it's clear that understanding these communities and how they change over time in a healthy eukymatoid system without disease will be important, because it will help us to establish not only the significance of the findings here, but also at what point taxa which may be linked to disease signs will start to become associated with eukymatoids. Our data tell us a little bit about the identity of the taxa that are present in our samples, but unfortunately we're still very limited on what we can say about the function of these taxa and their role in the development of disease. In the future, we can build upon the quite basic sequencing methods that we've used here to start looking at the functional capacity of the microbes associated, and also the response of the host itself, in order to learn more about the mechanisms behind the development of disease signs. We should also prioritise comparison of the microbiomes of cultivated and wild eukymatoids, which don't seem to have anywhere near the same problems with IID, either from our search of the literature or from the sampling that's taken place during GSS. So understanding the differences in the microbiome of wild and clonally propagated cultivated eukymatoids may give us some insight into this apparent difference in susceptibility.
Okay, thank you, Dr. Watt, for such a informative talk, especially as regard about the microbiome associated with ice disease. This is actually a very new work. Actually, I believe can be considered as first of its kind. Okay, now we'd like to move on to next speaker, which is Dr. Marit from Natural History Museum of UK. She will share with us the hidden world of green algae endophyte, diversity, relationship with hosts, and impact on aquaculture crops. The floor is yours, Dr. Marit.
Thank you. Okay, so I will open my presentation. Can you see this now?
Yes, we can.
Okay, good. So, yes, my name is Marit Bjor-Beckmo. So this is based on a review paper that I'm working on together with Juliet Brody at the Natural History Museum. So first of all, what are green algal endophytes? Endophytism, or to live as an endophyte, is a common phenomenon in several evolutionary lineages of green algae, and it occurs in a wide diversity of hosts in both aquatic and terrestrial habitats. For example, some green algae are known to live as endophytes inside single-celled eukaryotes, also called protists, such as, for instance, foraminifers, or let's see, I'll just find a pointer, and also ciliates, such as these. It's also known that some green algae live as endophytes inside animals, such as corals, and also cnidarian hydra, such as these examples here. And also in sea slugs and in amphibian eggs. They're also known, well known to form mutualistic symbiosis with fungi, forming lichens, and also, of course, inside seaweed, which is the topic of this talk. And they are known from various macroalgal seaweed hosts, where they often are considered to be pathogenic, causing harm on their seaweed hosts. So what I've started to do here is to construct a database. And the database so far contains endophyte host interactions from 40 scientific papers. And from these papers, there are in total 304 entries of interactions between green algal endophytes, or also some epiendophytes, which are associated with seaweed hosts. And these encompass interactions between 70 endophyte species and 147 different seaweed host species. And the majority of these entries come from morphological studies, about 64% of the data, but 36 of the interactions are also represented by molecular data in addition to morphological description. And some of my future plans for this review work, when I sort of consider that we are finished with the literature searches, is also to obtain an overview of the publication year and the geographic regions or countries that the various studies are from, to identify how this field has developed or progressed over time, and also to identify potential gaps in our knowledge. And this is a bipartite interaction network that I made. And I don't expect you to see all the details here, but you see that to the left, you have the seaweed host species, the endophyte species, and here you have the seaweed host species. And I highlighted some of the species which demonstrates that certain endophytes have a broad host range, such as the Ovella leptosheti, and also Ovella heteroclada. And others seems to have more sort of a specialist interaction with fewer different hosts, such as Ovella operculata. And another aspect that we have to consider is that certain endophytes and certain seaweed host species have been the subject of more studies, and hence this bias could potentially be one of the reasons that the diversity of endophytes associated with, for instance, Chondrus crispus is a lot higher than several other hosts. So when we look at this at the order level, it's a lot easier to see some patterns. So as you can see, the majority of endophytes belong to the Alveolus, and these interacts with both green, brown, and red algal seaweed hosts. However, it seems like green algal endophytes most commonly proliferate in red algal hosts, and 64% of the hosts in our literature review are red algae. And so, as I said, the majority of hosts belong to the red algae. And this is an ancient lineage that originated over 1 billion years ago at approximately the same time as the green algae. Most of the endophytic green algae in seaweed hosts belong to the Alveolus, as I said, and this is one of the core chlorophyte lineages. And the origin of an endophytic lifestyle in some Alveolus is unknown, but however, this group here, it went through a rapid radiation in this period in the late Neoproterozoic and early Paleozoic era. And this also coincides with the snowball Earth conditions and the Cambrian explosion. And some earlier studies have hypothesized that the evolution of multicellularity and macroscopic growth in the Olophyceans may have been triggered by the introduction of novel grazing pressure by animals in the late Cambrian and Ordovician periods. And we also now hypothesize that the evolution of endophytism in some Olophyceans may also have been triggered by the same factors as a sort of survival strategy during the glaciation periods and or to escape grazing. So this illustration shows a cross-section of a red algal host with the outer epidermis, cortex, and the inner medulla. It's not always easy to distinguish between what is an epiphyte and what is an endophyte, and where to draw the line between these two categories. Like many things in nature, we people or biologists like to put things in boxes or to have specific definitions which not always corresponds to what we see in nature. However, I tried to come up with some endophytes that can act as examples for the different scenarios shown above. And the first category is the sort of strict endophyte, where the green algae is only attached to the outer surface or epidermis of the seaweed host. And the second category is where the epiphyte forms rhizoid branches that doesn't penetrate the cortex, but it sort of spreads out and cover a larger surface area of the host. And the third category, where we can start calling the green algae endophyte, are the ones that penetrate into the cortex of the algal host, but perhaps without causing any or too much damage to the seaweed host. While the final two categories are characterized by green algal endophytes that penetrates the cortex and grows into the medulla, potentially also penetrating host cells. It is, however, important to notice that these interactions and the nature of these interactions, whether the endophytes are more or less harmless or if they seem to be acting as pathogens, can vary both due to abiotic and biotic factors, such as, for instance, in different life cycle stages of the seaweed host. For example, the gametophyte versus the sporophyte stage sort of tackle the endophytes differently. And the gametophyte of Chondrus crispus are, for instance, more resistant than the tetrasporophytes to infection by the filamentous endophytic algae Olvella operculata. And also different seaweed host species have, for example, different thickness and compactness of the cortex, and also different mechanisms for protection against potential pathogens, such as oxidative bursts, for example. Furthermore, several studies have shown that different parts of the thallus or the seaweed body such as the apex versus the middle versus the basal parts, are affected to different degrees by these endophytes. And abiotic factors such as increased temperature due to climate change can also influence the endophyte load. And all of these aspects leads us to the next topic I want to address, the concept of holobionts and dysbiosis. So first of all, what is an holobiont? So you have a seaweed, and a holobiont is an assemblage of different organisms which forms an ecological unit, often defined as the host organism, plus all of its microbial symbionts. And this involves bacteria, archaea, fungi, viruses, protists, and also endophytes and epiphytes, which live within and on the seaweed. So in other words, the holobiont comprises all organisms, from mutualists to commensals to parasitic organisms. And whether the green algal endophytes are parasites or pathogens or commensals without any direct negative effect on their hosts has been debated. However, it has been shown that some of the green algal endophytes have a negative impact on the growth and reproduction and the reproductive output of their seaweed hosts. And also the carrageenan yield has been shown to be reduced due to endophytes. For example, the Olvella operculata has a carraginolytic activity to degrade the carrageenan in Chondrus crispus. And in addition, endophytes and also epiphytes can facilitate secondary infections by pathogenic bacteria and potentially other pathogens. So a shift in the microbial community from a healthy to an unhealthy microbiota can have negative consequences for the host. And this is also what is called dysbiosis. Dysbiosis can be caused by, for instance, increased temperature due to climate change, which causes stress on the host. And this can potentially be particularly crucial for aquaculture crop populations with low genetic diversity, which makes them more vulnerable and potentially less resistant to pathogens spreading amongst individuals. And dysbiosis can also lead to an increased risk of disease from endophytes, because the host is, if the host is weakened, the initially harmless endophytes might sort of turn pathogenic. And in order to understand how seaweed hosts are affected by their microbial partners and how they are affected by sort of unnatural situations such as climate change and aquaculture, it's important to gain knowledge about all types of microorganisms associated with seaweed hosts, including the eukaryotic epiphytes and endophytes, in addition to all the other organisms living together with the seaweed hosts. So this was my presentation today. Thank you so much for your attention, and thanks to GlobalSeaweedSTAR. Thank you for inviting me to give this talk. And of course, thanks to UKRI and Global Challenge Research Fund.
Okay, thank you, Dr. Marit, for such an interesting topic. So green endophytes is something that is actually overlooked, but I think that it potentially can bring some harmful effect as well to the grower seaweeds. Okay, next, I would like to invite Dr. Janina Brekel from Scottish Association for Marine Science to share with us her topic on exploring the genetic resources of the seaweed crop Carpa ficus and Eucuma from a global perspective. Dr. Janina, please.
Good day to all. My name is Janina Brekel. I'm a postdoctoral researcher at the Scottish Association for Marine Science. It's my pleasure to present today the results of our project exploring the genetic resources of the seaweed crops Carpa ficus and Eucuma. Dr. Tanji gave a presentation earlier on genetic diversity of these commercially exploited seaweed species in Malaysia. This talk will complement this by adding some insights from the results obtained in other regions of the world using similar methods. The output presented here stems from great team efforts as reflected in the long author list here and institutions listed at the bottom of this slide. I will highlight specific contributions throughout my talk. There's a recognition that the ocean harbors a great richness and biological diversity that can be harnessed. In one such an example is the exploitation of seaweeds. As highlighted in talks before, seaweeds are a rich resource for a whole diversity of substances and products, including food or food additives, parts of cosmetics or medicine, animal feed or fertilizer. This provides opportunities for people to become economically active, specifically in coastal communities. Seaweed aquaculture also supports food security either by consumption of seaweeds directly or by its integration into multi-trophic aquaculture. The fast growth of the seaweed farming sector, specifically in the last 2 decades, can be seen here and underlines its relevance. The growth of the sector and the benefit from it rely on resilient and diverse cultivars and genetically diverse breeding populations. To develop and maintain these resilient cultivars, it's necessary to be able to tap from a pool of genetic diversity within cultivated and natural population of these seaweeds. In this talk, I will explore the genetic diversity of seaweeds of the seaweed crops, Capophycus and Eucuma. In the late 1960s to early 1970s, cultivation of Capophycus and Eucuma species has been initiated in the Western Coral Triangle in the Philippines and Indonesia. Based on the high success and growth rates, soon many other countries followed. Often seaweed strains were introduced from the farms in the Philippines to other parts of the world, as indicated here by the black arrows, for example to Tanzania, to Madagascar, Hawaii, India, and many other countries, where these seaweeds are not native. Thus, if we are looking at the distribution of genetic diversity of cultivars, we expect to see the pattern reflect this history of introduction. Until this day, cultivation is based predominantly on clonal propagation, resulting in a standing stock of genetic diversity. Cultivated species are Capophycus alvarezi, Capophycus striatus, Capophycus malaysianus, and Eucuma denticulatum. To obtain a baseline level of knowledge on the genetic diversity of Capophycus and Eucuma crops and their distribution, we sampled publicly available sequence data of the most commonly used marker at this moment from these species and mapped their global distribution as indicated here. We also reviewed for each species and each area how many different genotypes were detected. This is indicated by these pie charts, where every color points out a different genotype. As I mentioned before, the distribution here does not reflect their the natural distribution, but also includes the history of introductions. Yet we can see that the Western Coral Triangle and the Western Indian Ocean contain unique genotypes. While the overall data set that we compiled here is rather small and there's a bias towards cultivated individuals, we believe that the overall Eucuma and Capophycus diversity is yet undersampled, and we believe that there are more genotypes yet to be discovered. Therefore, for our project, we decided to tackle assessing the genetic diversity of Capophycus and Eucuma better, specifically the wild population, but also in the farms, where farmers might have taken up individuals from the wild and harbor and maintained this diversity in their farms. Our efforts went into designing a global sampling campaign. Our international team co-designed a sampling strategy. Our consortium sampled from wild population and farms, and we built up a joint database between different institutions to effectively share data and work jointly on this project. Extensive collection of herbarium specimens were built up specifically in Malaysia, the Philippines and Tanzania. Photos document the morphology of Eucheuma and Kappa-ficus specimen, and analysis of mitochondrial markers allows us to genotype individuals and to assess the level of diversity. As a result, we were able to sample in more than 83 sites for farms and 23 sites for wild populations, covering densely the Western Coral Triangle, the Western Indian Ocean, but also several islands in the Central Pacific. I will focus next on presenting some of the results with a specific focus on the Western Indian Ocean. First, I will illustrate an example where we discovered previously undescribed diversity that we have encountered in Madagascar and Tanzania. I'm showing you here a phylogenetic tree of the genus Kappa-ficus. It illustrates the relatedness of diverse genotypes within this genus. I'm zooming in on the top of the tree. I'd like to highlight here some genotypes that we detected in Madagascar, shown here. In Madagascar, we could detect 5 distinct genotypes that have not been described before. I'm showing you also here some exemplary images. Also in Tanzania, we could detect one undescribed genotype closely related to those in Madagascar, yet distinct. All these genotypes have only been detected in the respective regions and are thus presumable endemic genotypes to the Western Indian Ocean. They are closely related to the clade that contains the globally cultivated Kappa-ficus alvarezi, as indicated in red-orange. The detection of these genotypes provides also the opportunity for the development of potentially novel native cultivars for Madagascar and Tanzania. In a second example here, I'm outlining the results of genetic diversity encountered for the genus Eucheuma in Tanzania. In a sampling campaign led by GlobalSeaweedSTAR ESR Sadogrosveka, he encountered 3 haplotypes of the non-cultivated species Eucheuma platycladum. For Eucheuma denticulatum, a number of specimens have been encountered. However, all of these were determined as the cultivated haplotype introduced from the Philippines, and none of the specimens could be attributed to the native Western Indian Ocean haplotype of Eucheuma denticulatum that we had expected based on the baseline review and as depicted here. The disappearance raises concern for the status of the Western Indian Ocean native haplotypes. Reasons for declining numbers could be, for example, overharvest in the past, global warming, or competition with localized introduced genotypes. Going back to our initial question, what did we learn about genetic diversity of cultivars and wild stocks of Eucheuma and Kappa-ficus? I showed you one example that outlined undocumented diversity in wild population in the Western Indian Ocean and the genus Kappa-ficus. We could encounter haplotypes unique for the respective region, which provide a potential source of novel, locally native cultivars, and we documented with this region harboring valuable genetic resources. On the other hand, we had the case that cultivated genotypes could be detected outside in naturalized populations, as the example of Eucheuma denticulatum showed in Tanzania. This highlights the need of monitoring unintentional introduction of cultivars into wild population. Also, there's an unclear status of the presumable native haplotypes. With this, I would like to thank you. And
okay. Thank you, Dr. Janina, for such a very interesting talk that actually covering a wide sampling area up to 83 sites globally. Okay, and it shows that we do have novel haplotypes from different regions and different countries. So that's highlight there is a need for us to continue to work on, to conserve and to develop all these new cultivar, new novel haplotype, and through banking approach. Okay. Next, I would like to invite Dr. Martina from Scottish Association for Marine Science to share with us My Seaweed Look Weird, a participate web portal to increase knowledge on seaweed diseases. The floor is yours, Dr. Martina.
Good morning and good afternoon to everybody. My name is Martina Strittmatter and I'm a postdoctoral research assistant at the Scottish Association for Marine Science. I've been working for the GlobalSeaweedSTAR project for the last 2 years, and in today's talk I would like to introduce you to our participative web portal, which is called My Seaweed Looks Weird. Like any other living organism, algae can be plagued by diseases. Diseases in a very broad sense are any abnormal physiological or developmental condition that is caused by either abiotic or biotic factors, or even a combination of both. As already pointed out by the previous speakers, the cultivation of Capophycus euchyma is currently largely hampered by two main problems: the ice-ice syndrome and epiendophyte infestation. Likewise, other major seaweed-producing sectors face the same problems of diseases. And here prominent examples are the Neopyropia neoprophyra and Saccharina japonica industry in Asia, and the Agarophyton chilensis production in Chile. Comparable to animal aquaculture and agriculture, the intensification of seaweed cultivation will certainly lead to the increase of disease-related issues. But unlike in agriculture and animal aquaculture, disease diagnostic tools and biosecurity are still very much in their infancy and in urgent need of elaboration. In terms of phylogenetic origin, it is evident that algal pathogens and pests are of diverse origin and omnipresent, as it is illustrated in this tree here. We can find those pathogens and pests in all major lineages. Here we see, for example, omycetes and brown algal pathogens. Then there are fungi that infect macroalgae. We have red algal parasites and green algal endoepiphytes. And here you can see a phytomyxid pathogen. And of course, viruses and prokaryotes are also known to infect macroalgae. Such diseases are documented in wild and cultivated macroalgae. And nevertheless, the current knowledge on algal diseases is still very limited, which impedes a safe and sustainable development of the seaweed industry. Crucial data like pathogen diversity, host range, biogeography, and ecology are hardly available. In order to further and accelerate our current knowledge on seaweed diseases, we have recently set up a participative web portal which is called My Seaweed Looks Weird. I would like to invite you to visit our My Seaweed Looks Weird web portal, which can be accessed under this address here. So as I said, this is an online web portal that is currently available in 3 languages, in English, Spanish, and Portuguese. Observations of unhealthy looking algae can be reported via this web portal by filling in a short questionnaire. In the first section, you are asked some details about yourself, like your name and your email address. And then in the second section, you can specify your observations by detailing algal species, observed disease symptoms, GPS data, and so on. There's also a section where you can specify whether your reported observations have to be treated confidentially, and how you would like to be acknowledged for this submission and so on. Furthermore, we encourage the submission of material. Such submissions can consist of image material, which documents the unhealthy, unusual-looking alga, or even physical samples. My colleague, Dr. Pedro Murua from the Austral University of Chile, is in charge of samples coming from Latin America, and I'm in charge of samples coming from Europe and the rest of the world. At this point, I would also like to specify that it is best to contact us prior to sample shipment, as we can advise on storage conditions and fixation buffers, and we can also assist in the shipment of the samples. There are some macroscopic signs that can indicate the presence of potential pathogens in macroalgae. So first of all, there are galls and blisters and warts as shown here. Then we can also distinguish spots, bleaching and discoloration, lesion and holes, and distorted morphology. I haven't presented disease symptoms of filamentous macroalgae here, which of course are less obvious and require a first screen via microscopy, but they also show classical signs which are discoloration and host cell deformation. So once we have received potentially diseased samples, we can envisage several analyses which largely depend on the fixation and storage conditions of the sample. In the first instance, we will always perform a microscopy analysis, like for instance with a specific stains, in order to get first indications of pathogen structures and host cell reactions. If possible, we will then also try to get barcode sequences to identify the pathogen by molecular methods. However, this might not always be possible or might be difficult. For example, if the sample has not been stored in the right buffer, and if the pathogen density is very low and so on. Additionally, we can also try isolation, cultivation, and reinfections of putative pathogens. We will offer an in-kind diagnosis, and if possible, we will establish a report of our observations, which will then be sent back to the person who has submitted this observation. So our objectives of the My Seaweed Looks Weird portal are to encourage the participation and invite sample submissions, to accelerate the description and identification of algal diseases worldwide, to increase the knowledge on the diversity of algal pathogens in wild and cultivated macroalgae, and most importantly, to make this information available in an open access repository, which is called the Algal Disease Atlas, and which is currently in progress. So as I said, the Disease Atlas is currently in preparation, and it will contain taxonomic data of pathogens and host algae, as well as image material, like for instance different life cycle stages of the pathogen, microscopic and macroscopic disease symptoms, and so on. Furthermore, it will contain biogeographic data, literature, and protocols. The data that are going into the Disease Atlas originate to a large extent from already published literature, And we also aim to include relevant information and literature from non-English publications as well. Furthermore, data obtained from the ongoing projects GenieAlg, GlobalSeaweedSTAR, Alga Blister, and from the My Seaweed Looks Weird web portal will be included into this disease atlas. And the objective is really to publish a first version of this atlas at the end of this year. Our overarching goal with this atlas is really to make information available to a broad audience that is interested in seaweed diseases, and to provide scientific support and to give a platform for discussion and exchange on the topic of seaweed diseases. And on this note, I would like to thank our team here at SAMS and all collaborators of the projects GlobalSeaweedSTAR and GenieAlg, and I would like to thank you for your attention, and I'm
Great. Thank you, Dr. Martina, for sharing with us such an important initiative in developing My Seaweed Look Weird web portal. Okay. Next, I would like to invite Dr. Iona Campbell from Scottish Association for Marine Science to share with us a study on current perspective on biosecurity policy and practice in a global seaweed industry. Okay, Dr. Iona, please.
Hello, I'm Iona Campbell, and I've been working on the Global Seaweed Project for the past 4 years to bring an international perspective to the current status of biosecurity policy and practice in the seaweed industry. This presentation is a summary of the outcomes of various research activities aiming to understand biosecurity from both farmer and policymaker perspectives. With the growing global population and the future food security challenge, and a desire for many countries to develop blue economic growth, there are many national and regional plans worldwide for aquaculture expansion. And seaweed is already a significant contributor in volumes of aquaculture products globally. Just like many other production sectors, disease is a major bottleneck for sustainability and growth of the seaweed industry and in aquaculture worldwide. Each year, 20 to 40% of terrestrial global crops are lost to pests and diseases, and the aquaculture industry is no exception. Many sectors have already become limited by diseases and pests, and aquatic animal diseases now equate to approximately $6 billion of losses annually. And just some classic examples are sea lice on salmon farms in temperate countries and white spot syndrome in shrimp found globally. Earlier on in the project, we looked at the international policy landscape, and we tried to understand how this policy landscape either fits or where there are gaps with the seaweed aquaculture industry. And one of the main things was the separation in many of the policies between aquatic animals and plants, and that seaweed industry actually falls under the International Plant Protection Convention when it comes to international management of biosecurity risks. And since this work was published, there has also been some international initiatives regarding biosecurity in the aquaculture industry, and one of them being the Progressive Management Plan for Aquaculture Biosecurity led by the FAO, which is designed to assess and build aquaculture biosecurity capacity on a national scale. And another of those has been the recent publication on One Health in the management of the aquaculture industry as a transdisciplinary framework. As part of our research, we looked at the content of biosecurity policies for carrageenophyte industry, and evidence required to develop specific policies was often lacking for these to become specific and be more efficient for the seaweed industry to implement. There was also inconsistent terminology for how seaweeds were included in different policies, and also very limited guidance on who would be implementing different measures in the production of seaweed. Given the large-scale nature of some of these initiatives to build biosecurity capacity and policy and improve management systems, it's become quite timely now to understand where the seaweed industry might sit within them. This requires a certain level of understanding where the current capacity for seaweed biosecurity is. And it was for this reason that we designed a survey of farm biosecurity on an international scale to match the scale of these global policy initiatives. So we used a well-known survey tool called the Knowledge, Attitudes and Practice Survey Tool to help with policies and decision making with the practitioners in, originally it was used in public health and has been used in agriculture and food production systems since. And so we adapted this tool to assess farm practitioners in the seaweed industry. The survey was designed to assess and compare biosecurity of red algae production, Carrageenophyte farmers between Tanzania and the Philippines. And both of these countries were producing seaweeds for similar international markets with very different national value chains. And the survey was spread over the top 3 production regions within each country as examples of the productive and active areas of farming, which have also been reporting challenges due to pests and diseases. The participants of the survey covered just the farming system, so not the processing or the after the harvest of the seaweed. And it included from seedling to harvesting the raw crop, and it included farmers that owned their own farms and also farmers that were employees of larger farm systems. And in total there was 189 farmers who participated in the study with a participation rate of 96.6%. The survey also asked farmers if they had been provided biosecurity training. And what we found was that in Tanzania there was a diversity of providers of training, which ranged from non-governmental organisations, ministry departments, from government, other seaweed buyers and other farmers who were teaching, academic institutes. And then in the Philippines, what we found was most of the training came through the governmental departments, but there was also some training from NGOs and from research institutes. What we found was that farmers had good biosecurity knowledge but lacked the resources to implement the practices. For example, some farmers didn't have the ability to monitor their farms when they couldn't be accessed on foot or by boat, so when they were submerged at high tides. And that biosecurity policies in the seaweed industry could learn from these lessons and should accommodate smallholder and large commercial farms when on a large international scale. The survey also showed that there was sometimes a lack of clarity between what was generally just best practice in the seaweed industry and then what practices were specifically addressing biosecurity risks. And this came out in confusion over what biosecurity training had been provided to farmers. So messaging on biosecurity principles in farm management in national training programmes can be lost. From the questions around the practices, the practice scores in the Philippines were higher compared with Tanzania, but this was mostly due to the fact that Tanzanian farmers were very limited in what they could do because they were using off-bottom farming technique, which meant that farmers only had access during low tide and in daylight, so about a fortnight of every month. Farming techniques in the Philippines are highly diverse and access to the majority of the farms are independent of the tides, as many farmers can access them by boat, enabling the farmers to react quickly to the environmental changes compared to a fortnight on and a fortnight off in Tanzania. So just to conclude and put this work on policy and practices into perspective of biosecurity in the global seaweed industry, It's important that developments in national and international policies are adaptable to the different scales of production that exist in seaweed industry, such as smallholders and large-scale commercial seaweed employers. It's also important to compare biosecurity in the seaweed industry with other examples from aquaculture subsectors, which also have to incorporate the added complexity compared to terrestrial systems of an open aquatic environment in disease management. And that the results from this research coming from the angle of global policy and then global practices, we found that linking production, environmental knowledge and policy often lacks a distinction between what local knowledge and expert knowledge is, which are both extremely valuable when trying to tackle biosecurity. The evidence base to facilitate reconciliation and compromise between the different organisations of science and policy and farmers. There is a knowledge gap in the evidence base between these groups of people, and bridging the gap between the management of aquatic and animal plant health systems in aquaculture sectors are still largely divided and could do with being more connected if we're thinking about tackling seaweed industry on a global scale. And finally, there are common challenges faced by farmers internationally. For example, limited epidemiological knowledge from science limits what farmers can do to think about better management practices and techniques. And pest outbreaks related to climate change are often out of control for farmers. Local and socio-political contexts can influence the success of biosecurity interventions on an international scale. And finally, that international policies can be successfully implemented whilst maintaining sensitivity to the individual challenges faced by seaweed farmers on a local scale.
Okay, thank you, Dr. Campbell, for highlighting the importance of biosecurity for both national level as well as international level. Okay, next, I would like to move on to next talk by Dr. Virginie on social consideration to understand the sustainability of seaweed value chain. The floor is yours, Dr. Virginie.
Thank you, Professor Lim, and good day to all. Thank you very much also for this invitation to share our research. So together with my colleagues from Tanzania, the Philippines, and Malaysia, and from the UK, we've been focusing for the past 3 years on the social consideration of seaweed value chains in 3 different countries. As was presented by previous colleagues, this is a global comparison of how the stakeholders involved in the seaweed industries have been managing some of the risk that the seaweed farmers are currently facing. In this presentation, I will focus on 3 very brief points and trying to make the case as to what social sciences can bring to the aquaculture sector, particularly around the risk faced by seaweed farmers currently. By way of introduction, and as already explained by my colleagues before, there is widespread and worsening occurrence of disease and infestations. And this has been reported in all 3 countries where we've been working with. And there are also increasing reports of pest outbreaks, which are attributable to environmental shifts. One of the key findings that resonate a lot for our work on social science was that some of the species are unlikely to survive sudden increase in temperature above 36 degrees. And yet we are living in a world that is going to face an increasing number of hot days based on the latest findings from the IPCC report on climate change. While our colleagues focus on what it means for the growth of seaweeds and the impact on biosecurity, we've been largely focusing on what this means for smallholder farmers for their daily activities. The question that we asked was, what do they do to face disease outbreaks in addition to many other constraints on their livelihoods? And the assumptions that we work on was that not all farmers are equally equipped to adapt to risk, because this is largely influenced by their access or lack of access to support, such as equipment, trade regulations, biosecurity knowledge, social networks, as well as bargaining power. So we relied on social science because it allowed us to challenge the assumption that only working on seaweed as a crop will automatically improve seaweed farming. So we really much focus on the human factor here. And so that meant we mobilized research tools that focus on human interactions. So some of our tools include what we call key informant interviews, also discussions as a group called focus group discussion, as well as participant observation and thematic analysis. With these approach, we explored in more details what happens for seaweed farmers affected by disease outbreaks in different places. We also asked the why question, why some cultivators, for example, in Tanzania, stop farming seaweed altogether, while other farming communities in Zanzibar keep growing and flourish. We also asked how do farmers deal with the different risks that they're facing? How do these strategies might change in different contexts? Because that will help us build a more nuanced picture on what are the current risks faced by seaweed farmers and what are the current strategies to deal with those risks. The second importance and approach in our work was to locate seaweed farmers within their wider socioeconomic context. While many of our colleagues worked on how the environment, environmental parameters of seaweed farming in fact affect the growth of crops, we looked at the societal factors from social context factors to political dynamics, as well as interactions within and between different regions. So this is part of a context analysis, but we also looked at the different segments of the value chain of seaweed, particularly the carrageenan value chain. And that means looking more closely at the relationship between farmers with collectors or traders, but also between traders as well as buyers, processors, or exporting companies, and with the role of local authorities as well as national authorities. And this value chain analysis combined with context analysis enable us to map the different stakeholders that are involved in the production and the transformation of seaweed, but more importantly, to examine where there may be hurdles or opportunities for sustaining seaweed industries. So in Malaysia, my colleague Azam already presented how much a closer understanding of who a farmer is enable us to better estimate who produces most of the seaweed. And this has huge implications for developing the sector. To summarize the benefits of an approach where we combine social science with the work of our colleagues in the natural sciences is that we are able to build a better understanding of the stakeholders' socioeconomic situation and the political context. Second, we better acknowledge that farmer status influence whether their presence and production are officially counted or not, and if they have access to government schemes to obtain permits, certification, or training. And all of these are requirements to enable the growth of the sector. What we found in Malaysia was a potential disconnect between the assumptions and the reality of farmers' experiences, because we needed more nuanced understanding of who the farmers are and what challenges they face. And this could potentially limit institutional efforts to share knowledge and best practice in biosecurity, as emphasized by our colleague, Dr. Cecilia Combe, among seaweed farmers, but also between seaweed farmers and the other stakeholders that they are trading with. And this overall would contribute to secure seaweed production if we better address those hurdles. The last point we wanted to make is that throughout our work in the past 3 years, we try to apply a gender lens. And a gender lens or a gender responsive approach is a way to recognize the differences between men and women who work in the seaweed value chains. What we found, and that was already published many times in the literature, was that men as well as women are present at all levels of seaweed value chains, but they sometimes occupy very different roles depending on the gender division of labor in some contexts. So for example, women are mostly associated with pre and post-harvest activities, while men dominate downstream activities and therefore the higher returns. But the main point here is that farmers do not work in isolation, and particularly in informal context, seaweed cultivation often relies on the less visible work of other family members, and this less visible work is also often unpaid, and that's predominantly performed by women. Seaweed trading is also influenced by different social relationships between producers and buyers, and these need to be also understood in order to understand if biosecurity knowledge can be shared with people who need it the most. One of the key findings from the work of our colleague GCU in the Philippines is that some gender stereotypes result in the misrepresentation of women in seaweed farming and seaweed trading. Social and economic structures are sometimes inadvertently favorable towards men, whereas the women's access to resources and opportunities are deterred by traditional beliefs and practices. They often have fewer opportunities to participate in policy discussions and negotiate their economic position comparable to men. So policies and policy-making process therefore tend to be biased towards men because of the failure to take into account women's actual contribution to the seaweed industry and the unique challenges that they may face. To conclude, a better understanding of who farmers are and the challenge that they face, allowed by relying on the combination of social sciences with natural science, is a key opportunity to better address some of the sustainable development goals, starting with SDG 5 and 8 on gender equality and decent work and economic growth, by allowing all farmers to have the basic needs met through appropriate services, resources, and securing their income. They should also all benefit from equitable support and strong formal and informal networks. And our work in Malaysia, the Philippines, and Tanzania show that it's not really the case at the moment. Second of all, to address SDG number 13 on climate action, biosecurity policies and policies around seaweed farming have a potential to contribute to risk mitigation measures to help the sector adapt to the increasing environmental shifts that they're facing in order to protect the assets of the farmers. Also, appropriate information could help value chain stakeholders to identify and apply adaptation strategies. And then lastly, this would overall contribute to SDG number 14 on life below water by supporting more sustainable ecosystems that are effectively managed to preserve biodiversity, also for the use and the benefit of farmers and their communities. And with this, I thank you for your attention and go back to the main floor. Thank you very much.
Thank you, Dr. Virginie, for sharing with us. I think that Dr. Virginie talks has actually helped to wrap up the whole session and the work of GlobalSeaweedSTAR that actually emphasizes that there's a need of equal emphasis for social, economic, natural science, biosecurity for the sustainability of aquaculture industry, okay, based on our study. So now I would like to move on for the Q&A sessions. And to start off with the Q&A sessions, there is one question that is to Dr. Pong that asking, what do you think about the sea microbiome will affect the development of ice syndrome? And probably after that, Dr. Watts can also contribute to some of the questions. Okay. Dr. Pong?
Yes, thank you, Clement, for your question. Right, so as I have typed in the chat, we actually do think that the microbiome in the seawater do affect the development of ice syndrome on the seaweed crops. So as I was saying that during our study in the Sentinel farm, we collected samples from the water and also compared it to samples of healthy and diseased Carpophycus plants. So the analysis is ongoing, and probably Georgia can tell us more about that.
Dr. Georgia, would you like to explain further based on your finding?
So, sorry. Yeah, so we, the Sentinel farm, as I explained it in my talk, we did collect the water as well. And I didn't include it in my talk because kind of there was a limited amount of time to kind of talk about things, but we did find the same groups that we are associating with the diseased tissue were also kind of present in the water. It's difficult to kind of place that into a context. So we know obviously that those groups are in the water. We don't know what it is that influences them settling and becoming part of the microbiome of the diseased tissues. And obviously when we're growing things in the open ocean, it's quite limited in really what we can do in terms of stopping them from settling. So we can kind of look at these things and we can understand where these things are in the environment. And the next step from that is understanding at what point they start becoming a problem with the seaweed. So we need to look back at where we've got this whole temporal time set that I'm working on now, just looking at where these potentially problematic microbes actually become part of that microbiome and understanding what it is that's changing within the environment potentially or within the host itself, and linking that back to where we start to see these disease signs. Does that answer the question?
Yep, thank you, Dr. Georgia. Okay, so now I would like to move on the second questions that's directed to Dr. Tanji from IAN niche. So his question was, is there evidence support for the Carpophycus cultivar genetic decay hypothesis? Or if so, what mechanism may be involved? Dr. Tan, could you please address this question?
Thank you, Dr. Ianish, for your question. Thank you very much for your contribution to the entire seaweed industry as well. In terms of your question, it's a very interesting one. It is still hypothetical. Based on what we know, most of the analysis were done on simple organisms. So basically viruses, bacteria, and some yeast. Typically organisms with short life cycles. So in this case, we do not have actual case or proof showing that it happens to, for example, plants or seaweeds or fungi. Yeah, that is based on what we know, but we do know that spontaneous mutation is always happening. So it is a slow process, so we still have time. However, what most people tend to overlook is that exposure to environmental stresses may actually quicken the process. Okay, so that's one of the reasons why we have to work hard to develop new cultivars, introduce sexual reproduction in order to better prepare for changes like climate change and so on. It's a very good question. Thank you.
Thank you, Dr. Tanji, for addressing the question. So the next question is addressed to Dr. Marit. Okay. Can the epi-endophyte continuum may also be a result of how a thallus is vulnerable to its host? Marit, would like to answer this?
Yes, I think that, as I sort of said in my talk as well, that I think there are like several different aspects, both abiotic and biotic factors that affects whether or how this endophyte or epiphyte, how it sort of interacts with the host. So yes, I think also the sort of like the structure of the thallus also affects how an epiphyte or endophyte acts with a specific host. And also, as I said, it has also been seen that the different life cycle stages of the host also affects how the endophyte sort of interacts with the host as well.
Okay, thank you, Marit, for addressing the question. Okay, we have another question that is actually quite interesting from Algeria. She's asking about, is there any recommendation or suggestion on how we can reduce the epiphyte invasion in the seaweed farm? And Dr. Flower, would you like to answer that?
Yes. Since the problem of diseases is mostly based on impact of climate change, I think the best way is to do the good farming practices. Issues like harvesting the seaweed, early detection of disease signs, and harvesting the seaweed when it is not yet affected too much. Issues like deepening lowering the farm to deeper waters, to higher depths when the disease the disease signs are observed. If you lower them to higher depths, the diseases will be reduced. And things like choosing the seed, the best seed that has no diseases and so on. So I think the best way is to do some practices that can lower the risk of spreading of the diseases.
Okay, thank you, Dr. Flower, for addressing that. Dr. Pong, would you like to add on?
Okay, thanks, Prof. Lim. So I think prevention is much better. So what we can do is, from the output of our research, we are hoping that we could develop rapid test kits that can be used in the farm so that the farmers can use it to detect or have an early detection of whether the pest occurs on the seaweed crops before they grow it out into the farm. And this will cause a spread of the epiphytes into the rest of the crops. So yeah, and as Dr. Flower pointed out, it is very important to carry out biosecurity measures such as quarantine of the seedlings before you outplant them into the farm. Yeah, I think that's all, Prof.
Okay, thank you, Dr. Pong. Okay, there is one question directed to Dr. Cecilia, which is asking how biosecurity is controlled or managed in terms of household use and sanitization system for the floating house. So of those living in the vicinity of the farming area. Okay, so Dr. Cecilia, can you address that?
Okay, yes. My answer is about the how to manage the risk. So if you living in the not in the coastal area, but in the far from the in the middle of the ocean, I think it's better to still do the good agriculture, good aquaculture practice that we address in biosecurity procedure. But to add on about the risk that can contribute to worse environment system, I think in here that proactive from the government also we need in here. In our standard operating procedure is also addressing about the risk assessment. So if we cannot see that the risk, we cannot clean or remove all the risk, we just manage the risk. I think in here we just need the control on manage the environment system with the risk assessment. I think that is from my side. Thank you.
Thank you, Dr. Cecilia. So probably we move on to the one final questions that is asking about from Dr. Clement Wong, asking about is it possible to use biocontrol agent or beneficial marine microbe be applied during seaweed cultivation to suppress disease infection?
Okay.
Yep.
Okay. Yeah, so as I was responding to Clement, I think this is possible possible, but you have to be very careful and to conduct a comprehensive assessment of the effects of these biocontrol agents or beneficial marine microbes. What are their effects to the wider environment? Because we need to make sure that we're not introducing anything that is foreign or that will affect the ecosystem as a whole, because unlike the terrestrial plants where we have more control of the treatment being applied within a spatial boundary, so things are more challenging to be manipulated in the open sea. So yeah, you have to be very careful when you want to conduct a trial on this in an onshore hatchery, and you should make sure that the water is properly sterilized before discharge. Thanks, Prof. Lim.
Okay, probably I will take one final questions that are posted by Christine Rollin. It's actually a general questions that all the panelists, if you would like to answer, you can raise up your hands or highlight. Okay, what capacity or resources and limitation have you found available for setting up seed bank? Where would this be placed? Research institute, community, and farmer. Anyone would like to attempt to answer this question? I think this is quite, I will answer first to address this question. Based on the 2 days talk, we have highlighted, indeed, there is a need, urgent need actually, for us to set up biobank or seed bank for sustainability, availability of the cultivar. Okay, so I think the most important thing is we need my personal opinion, I think that it needs to be carried out by the government assigned stakeholder that is in charge for seaweed cultivation, because this needs a continuous sustainability that provide the seed. But in order, before we can set out the seed bank, there is a need for research, further research, before we can scale it up in a large quantity. So this is the thing. So, and I think is as regard about the place to be set up, is always the local authority that is close for the access for the farmer. Yep. So anyone from the panelists that would like to add on this aspect? If there is none, I think I would like to conclude and would like to thank all the speakers for sharing with us, all the 10 speakers for sharing with us such an inspiration, informative presentation resulting from the GlobalSeaweedSTAR project. So I would like to give a closing remark, okay, for this event, okay. So I would like to thank all of you, all the speakers, all the panelists, as well as all participants for joining us for this 2-day event on the GlobalSeaweedSTAR sharing best practice for seaweed aquaculture industry. I would like to record my sincere appreciation and acknowledgement to the funding agency, the GlobalSeaweedSTAR program that is funded by UKRI under the GCRF program, and all the collaboration institution. Okay, so I would like to acknowledge the program leader, Professor Elizabeth Cocktail-Cook, who is the program leader, as well as also the work package leader for WP2 that's working on biosecurity. And also special thanks to Juliet Brody, Claire Gachon, as well as Louis Section 4, their guidance to Malaysia teams, okay, during the implementation of the project. And then also special thanks to all partner institutions, as well as to all the UK PDRA, that actually they are the one that is actually closely working together with our ECR to help out, and also all the support given by management team of SAP, and also the ECR in Malaysia And most of all, we are very, very grateful to the local stakeholder in Malaysia, especially the Department of Fisheries Sabah, in helping us to conduct the study. Okay, and some of the impact and the high, as regards about the study that I would like to share is regard about the, I would say that this is the very first kind in Malaysia on study on the seaweed aquaculture, which include the science as well as socio-economic. And some of the research highlight that I would like to highlight that we found through the study is list of the epiendophyte. And in fact, many are this first time being known in the science. And this provide a future direction in developing molecular tool or rapid test kits for early detection in cultivar. And we have found that there's large pool of wall population that can be developed as new cultivar, and this had to be incorporated with the establishment of the biobank. And the importance of biosecurity application that can potentially reduce disease and pest infestation up to 75%. And based on the value chain analysis, there's a need to enhance the downstream activity, incorporating aids for migrant. And in short, okay, this is just somewhat like, but I would say that in short, GlobalSeaweedSTAR program definitely have made significant impact and leave its legacy toward the euchymatoid industry in Malaysia. And some of the highlight that I would say is that throughout this 3 years, so far we have published 2 books. As regard about journal, we have 6 Malaysian lead author, 3 UK partner leads co-authorship, as well as there are many more that is under preparation. Of course, there are a lot more impact that is brought through the GlobalSeaweedSTAR project. But today I'm not going to highlight everything, but I would like to invite you all to join us during the GlobalSeaweedSTAR online finale, which is going to be held on the November 10th. I hope you all can save the dates. And for further information, please contact globalseaweedstar@sam.ac.uk. So with that, I would like to thank you, all of you. And also I declare the close of this 2-day seminar. Thank you.